Hey, good afternoon, everybody. Welcome to our second lecture. In this particular lecture, we're going to take a look at phylogenetics and cladistics as this pertains to the history of life. So we're going to talk about how organisms are categorized based upon morphologies and what we call synaptomorphies. So these are going to be shared derived characteristics, even characteristics that are actually evolved on there. So here is the generalized outline for you guys, or the study guide for you. I think I have most everything on there, and if there's something that's not on there that I cover, you probably should know it. And then if there's something that's on the study guide that I didn't cover, shoot me an email and I'll cover it, or I will probably just not worry about it. But one of the big things that I want you guys to understand on this one is how to draw cladograms, how to interpret the information and draw the cladograms, and or a phylogenetic tree on there. Okay, so with that, we're going to move on and start talking about some stuff. So in this portion of chapter 20, what we want to look at, or what we're going to look at, is phylogenetics and systematics. And systematics is essentially the study of how biological diversity and evolutionary relationships, how are these interlinked. And phylogenetics now is looking at the similarities and differences amongst the different species. And one of the things what we could do is we could take the evolutionary history of the species or a group of species, and we can propose these particular phylogenies based upon the systematics on there. Yeah, I know that seems kind of confusing on there, but one of the things I like to do is I like to put this image up right here, which you guys have been looking at for about 45 seconds. And in this image, my question to the students when we're in class is, hey, what is this? Is this a snake or is this a lizard? And so, you know, everybody's like, oh, it's got to be a lizard because you're asking that question, right? I said, well, based upon that, what makes it a lizard? So most folks, when you first look at this, they're going to say snake. But this is actually a legless lizard. And the reason why you could tell that it's a legless lizard is you can kind of look at the tails a little bit different. Okay? But what gives it away is you see right here, there's an ear hole. I call it ear hole because, you know, that's the old, you know, you used to want, when I play football, you used to want to ear hole somebody, right? And then that's how you get a decleator on there. But it's the external auditory meatus. And that's what that is right there. And so you can see it in lizards. So if you go outside, you catch one of those little blue bellies, those western fence lizards that are running around, right? You can actually see, you can see the tampon membrane. You can see the external auditory meatus. Lizards have eyelids. Snakes don't. So you will always lose a blinking contest to a snake. Right? Although the snake won't know that. All right. But this is actually a legless lizard. We actually have legless lizards locally. They're much smaller than this one. There's actually a species that was more recently discovered. More recently, like within the last 10 years, in an empty vacant lot near LAX. So that was actually pretty darn cool. But we have them like down in San Diego, down into Baja, California, that area. So this is actually a lizard, not a snake. This is a legless. And if you ever watch Indiana Jones, right, and he falls into that snake pit, if you look carefully, there's a lot of snakes in there, but there's also a lot of legless lizards on there as well. So when we take a look at all the living organisms on this planet, we're going to put them into groups, and when we call these groups, we oftentimes refer to them as a clade, right? And a particular clade, I'll spend more time talking about it, basically consists of the common ancestor and all of their descendants on there. So here are all these organisms on the planet, but there are three big major groupings that all the organisms are actually placed into. Two of which are for the prokaryotes. That's going to be bacteria and archaea, and you can kind of see them on here as well. And then the one group that we're in is eukarya. And what makes us different is that we have membrane-bound organelles. Okay? And we also have histone proteins. However, archaea, they also have histone proteins, but bacteria doesn't. But these two are prokaryotic in origin. So when we take a look at this tree of life, one of the things that we're going to see is we're going to see kind of a couple ways on how to draw them or how they can be drawn. So in this bottom images that you see down here where it says rooted and unrooted trees so when we take a look at an unrooted tree, what we're really looking at is we're looking at relatedness for organisms that are pretty related to each other, okay? But we're not looking at common ancestry. So when we come over here and it's rooted, so at each one of these points right here, that's referred to as a node. So that little branch is a node right there. And a tree that's rooted, now what you're looking at is, so for example, I'll come right over here. So this is archaea, this is eukarya, this is bacteria, right? So I'm going to come right here to this node right here. And right here, and I'm going to take a look at, if I take a look at this node, and I take everything that's in red right here, these are the eukaryotes, okay? These are the eukaryotes right here, and they're rooted right here. So we'll take this common ancestor and all of its descendants. That's what it means to be rooted on there. Whereas in an unrooted tree, you're just looking at, what do they have in common? And this is how we're going to group them together. Okay? So one of the things that we're taking a look at, we have a single ancestral lineage, right, versus no common ancestors. Okay, so common ancestors, no common ancestors right there. So when you take a look at a phylogenetic tree, one of the things that we're going to do is you guys are going to draw some trees, and each one of these branch points right here, that's going to be a divergence. Right? There's going to be a common ancestor, and then here are the descendants. So these are oftentimes referred to as a sister group. Okay? And so with the phylogenetic tree, you're looking at relatedness. So here you can see an example. Everybody's got a vertebral column. Do they have a vertebral column? Yes or no, right? Well, in the case of the lancet, they don't have a vertebral column. They do have a rigid support structure made of a cartilage. Okay? Lamprey also has a rigid support structure made of cartilage, but it's kind of more considered a vertebral column now. All right? And so we'll take more look at that. We'll dissect one of those. And then, hinged jaws, yes. This one does not. Right? Do they have legs? Well, depending on which fish that you're looking at, they could have legs, so to speak. But it should be tetrapods. That's a better, 4 walking legs is better, right? Amniotic egg or amniotic membrane, right? Hair, right? So this is how you end up with this. So typically what you do see is you do see the trees. They all should come out here to the same line right there. So when we take a look at a phylogenetic tree, one of the things here that we're looking at is we see that this tree is rooted. The reason why this tree is rooted because there's a common ancestor. These are all the descendants. So when you take a look at a taxon is essentially a rank that's given to a particular organism or a group of organisms. So you guys remember the taxonomic classification: domain, kingdom, class, order, family, genus, species on there? Each one of those levels can be considered a taxon on there. So it's a categorical ranking, so to speak, on there. Okay, and so when we take a look at this, we have this phylogenetic tree is rooted. So here's a common ancestor that gives rise to taxons A through G. And when we take a look at each one of these branch points right here, here is a divergence. And when you typically see this divergence, what you're going to see is where it branches dichotomously, right? So there's going to be 2 branch points. In the case of this one right here, right, you actually have 3 branch points. So there's a little bit of uncertainty right there. You're not sure how these 3 are more related together. So for example, D, E, and F are grouped together as sister taxa. But this is an unresolved node because you typically don't have 3 points coming out. You always have 2 points coming out of there. Okay? So notice that we have 2 points. We got one going over here and one coming over here this side. But notice at this point, right, one going over here and then over here. So then at this branch point, B and C come off of there as well. So when you look at sister taxa, you're looking at the groupings of which. And so these 2 sister taxa that's highlighted right here, B And C, here's the common ancestor, and here is the taxa right there. Right. And so these two, because they're coming off of that same branch point, they're referred to as sister taxa. Now, if we want to include A on there, then we got to include this branch point right over here. Notice that taxon G right here is all by itself. So here's this branch point, right? So these guys are all, if we think about it, these are all grouped together, but G's out here all by itself. This is going to be our outgroup. This is the basal taxon. This is one that has some features that are in common, but it's not the main group that we're actually looking at. So when we typically look at vertebrates, the lanceolate is oftentimes used as the outgroup. Even though they are a chordate, they don't have a vertebral structure made out of either bone or cartilage. They do have what's called a notochord, which is cartilaginous in material. What this phylogenetic tree also shows you is here is the past, and here is the present. And one of the things that you'll notice is that all these taxa are all right in the line right there. And what that means is that these are all surviving members. I have some a phylogenetic tree later on that I'll talk about where the taxa line, taxa is plural, taxon is singular. That a taxon line does not make it all the way out to the very end where everybody else is, and that taxon is an extinct taxon. Okay. All right. So here is our ancestor. These are all the descendants. So with taxonomy, most of you guys are familiar with taxonomy. Right? So here's this term taxon. You'll hear it used all the time. So when we take a look at this, each of these domains down here, each one is considered a taxon. And when we take a look at this, these are very inclusive. Domains are very inclusive. When we get down to a kingdom, it tends to be a little bit more exclusive, but it still includes a lot. As we continue all the way down into the specific species, now we're super exclusive on there. So you guys should all know domain, domain, kingdom, phylum, class, order, family, genus, species. And whenever you get to the species name, all right, the genus and the species is always italicized, the genus is capitalized, and the specific epithet is in lowercase. And you cannot write just partus on there, because if you just wrote partus, what are you talking about, right? So right now, I know that we're talking about a jaguar. Okay, and so Panthera is the genus, because there's another genus that's Felis. Right, your kitty cat is in the genus Felis, as is the mountain lion, as is the mountain lion on there. The bobcat that we have locally is in a different genus that's in Rufus. Righteous. Right, so domain, kingdom, phylum, class, order, family, genus, species on there, and then you might have some subspecies on there as well. Right. So Carl von Linnaeus gave us this binomial nomenclature. So this two-name naming system, that's the binomial nomenclature. So we tend to use scientific names, so then that way it doesn't matter where you are in the world, you know what everybody in that locality, if they know that particular scientific name, they know what you're talking about. So for example, I'm going to use the mountain lion. The scientific name for the mountain lion is Felis concolor. So out here, most of us call the mountain lion. Depending if you're in Colorado, if you're in the Rockies, they might call it a cougar. And if you're down in Florida, they call it the panther. Right? Some places call it a puma. So maybe if you're down in Texas or New Mexico or Mexico, that area, they might call it puma. The high school I went to in Georgia for, not even a full semester, till we moved out to California, we were known as the catamounts. Right? That's another name. That's a lesser known name for Felis concolor. Right? So that's the mountain lion on there. Right? So that's this process. And so one of the things, just to kind of illustrate, right? So if we're up here in the domain, it's pretty inclusive. Everybody, come on! As long as you're a eukaryote, you're in the boat. But when we get down more specific to a kingdom, now we're going to exclude a lot of things, right? So if we're in an animal kingdom, plants are going to get excluded. Fungi are going to get excluded on there. Protists are going to get excluded on there. Chordates, okay, Now we're talking about animals with a rigid support structure in the back on there. So no more insects, right? No worms, no clams, no crawdads, right? All those different things on there. Now, of all these chordates, notice that we got fish, we got a lot of mammals right here, right? Oh my God, those are all pretty much all mammals right there, right? So, but we have reptiles, we have amphibians, those are all chordates on there. Birds are actually reptiles. But now if we go with mammals, what makes a mammal a mammal? That's right, mammary glands. So what we're looking at are who has homologous structures. Everybody in here, as they're being grouped together, they're going to have homologous structures that are of the same embryonic origin. So if we take a look at a bat's wing and a bird's wing, those are homologous, but an insect wing would be analogous structure. They're used for the same features in flight, but they're developed differently. Oh, here we go. Here's the bird wing and the bat wing. So this is a homologous. These are homologous structures, same embryonic origin. They're constructed differently, but then an insect wing, it would be an analogous structure on there. Okay. Another way that we can look at evolutionary relationships in terms of how organisms are grouped is looking at molecular biology. And so there's a lot of things that were once thought to be different species are actually now grouped together in the same species but different subspecies. So there's a species of rattlesnakes in Costa Rica. So and it was divided before, like in the middle, and so now because of molecular genetics on there, they're actually placed more closely together as two different subspecies, as opposed to two different species on there. All right, so when we take a look at a taxonomic tree, phylogenetic tree right here, right, so my question to you is, which taxon on the tree below is most closely related to taxon C? So you can see, think back, right? So this is a, first thing that comes to mind should be, this is a rooted tree, right? We have a common ancestor. These are all the descendants. So A right here, notice that it's all out here on its own, on a little island. So A must be the, that's right, the out group. B, C, and D are the in group. And so now our question is, who is the most closely related to C? Right? And that's going to be, that's right, that's going to be D, because there's going to be a characteristic right here that C and D actually share together. Right? And so remember, all these branch points right here, those are speciation events. So what does that mean, speciation event? So a speciation event is when you end up with new species developing from within what's already existing. So remember, there's geographical barriers that can establish that. Because I only primarily talked about allopatric speciation. I didn't really talk about sympatric speciation. I probably should have talked about sympatric speciation. So sympatric speciation is a speciation event where you're within the population, but something changes enough where, even though you're in the same locality, you're not physically separated. There's enough changes where you no longer have gene flow come into play. So like with trees, and oftentimes trees and plants and so forth that don't move, they oftentimes will have soil conditions, maybe a pollinator, things like that that are slightly different where they can exchange gene flow. Now with animals, it's a little bit typically harder to understand. And so what I'm going to do is I'm going to insert a slide right here, and I'll talk about sympatric speciation with you guys. And I'm going to talk about the cichlids, these fish that are in Africa, and they're pretty darn popular tropical fish for tropical fish hobbyists on there as well. Okay. All right, so D was the answer that you guys were looking for. I think that's what you guys told me, right? So before I talk about sympatric species, I want to finish train of thought over here. So which taxon on the tree below is most closely related to taxon C? All right. That was the question I asked you guys earlier. So this point right here, right? Common ancestor to A, B, C, and D. So right here at this point, this is going to be the common ancestor to B, C, and D. And then right there at that point, common ancestor to C and D right there. So when we take a look at phylogenetic tree and clades, one of the things that I want to reiterate is a clade is a common ancestor and all of its descendants. Right, so you can see right here in this green box right here, here's a clade. You have a common ancestor and you got all three descendants. This is a clade because there's a common ancestor down here, and then there's only one group of descendants on this branch. Okay. Now, if I come all the way out here at this point, then that would have to include these guys. Now, so if we kind of I'm gonna kind of draw some boxes around there, and that's going to make this nested. And so one of the things here when we talk about these clades, the common ancestor, all of their descendants, this is what we refer to as a monophyletic group. And that's what evolutionary scientists, that's what all these researchers are trying to do. They're trying to look at, here's the descendant, who are all the here's the ancestor, who are all the descendants off of there. Okay? And so one of the reasons why you want to do this is that way you have a better understanding of the relatedness on there. So if I put these blue boxes all the way around, because I have all these this one blue one right here, okay? What I want to look at right here is these are all monophyletic groups, each one of these. Each blue box represents a monophyletic group independently. So we're looking at 1, 2, 3, 4, 5, 6, 7, 8 monophyletic groups, because we're looking at their descendants and with that common ancestor on there. Now, I can make this nested, so you notice there was already that green box I was circling around, so I just put another green box around each one of these. This makes it a nested set, and as a result of that, each one of those green boxes is a monophyletic group because you have a common ancestor and all of their descendants on there. So if you're looking and you're thinking ahead going, wait, can you put another box, right? So we can put another box around each one of those, and that would be a monophyletic group as well. Right? So now what we have is we have what's called a nested tree, right? A nested set on there. So notice that there's a lot of characteristics that all these members on this particular tree, this particular branches actually are exhibiting. And so when we take a look at this tree down here, right, this is not considered monophyletic, and this is not considered monophyletic. On the one over here on the left, you're taking clades from different groups, so this is why this is not considered a clade, because you're not including the common ancestor, right? In order to get the common ancestor for these three right here, you have to take the bigger box. This is that red box that we have right there, and this is not a common ancestor because now you're excluding one of the members on the tree, right? So when we take a look at the classification of phylogeny, so here's what we have. We have Panthera pardus, we have Taxidia taxus, Lutra lutra, Canis latrans, Canis lupus. All right, so we got wolf, coyote, otter, badger, and the leopard right there. The leopard is our outgroup because this main group, what we're looking at, even though they are all carnivores, so this is a taxon, this is a taxon, this is a taxon, and these are taxa. Or I should say these are taxa on there. Taxon is singular, taxa. Carnivora, right? Felidae is the outgroup over here, and these guys are the in-group on there. So here you can see their relatedness. On this phylogenetic tree for elephants and their ancestors, what we're looking at is here is the past, and here is the present. And so if you're looking at this, we have 3 living elephant members, but what's grouped in here with them are a small little critter called a hyrax. Oh, rock hyrax is always so cool. I was able to see one when I was in Africa back in '92. It was so far away, but those guys are cool. But those guys are related based upon some particular structures that they share in common. And then manatees and the dugongs and sea cows all fall in that same category as well. But so notice that these 2 up here and these 3 make it to the line, so these are still living. Notice that these lines are a lot shorter in length, and so what that tells you is that these organisms are extinct. And because the length of the line in this phylogenetic tree is important, because we have a timeline on here, so we actually now know the length of the line, the closer they are to the present day. So like the mammoths, right? They're pretty darn close, but if we come back even further, some of these other ones they don't make it, and so that what that means is they were extinct for a heck of a lot longer period of time. So my question to you is, which taxon on the tree below is most closely related to A? Did you guys say B? Right, so notice that's that first branch point, right? That's going to be the closest. What can you tell me about A? A is the, right, outgroup. Which two taxa on the tree below are the most closely related to each other? A, B, C, or D? Or I should say A and B, A and C, A and D, or B and C, B and D, or C and D. Right. That's right, C and D. There you go. Nice job. And so phylogenetic trees, they might not be used for a whole lot of different things. You know, I'm going to do this, you know, so I get it. And so, but I will talk about briefly, you know, why should you understand phylogenetic trees towards the end of the chat. And so phylogenetic trees was actually used to discover some whale meat was being sold in a Santa Monica restaurant. And so the parent company is Typhoon. I forget the actual company's name, but the two Japanese chefs, they actually were illegally selling whale meat in the U.S. and serving it over there at a Japanese restaurant in the Santa Monica airport. And so what they did was they took samples and then they compared it to the known DNA sequences. And they were actually able to deduce which particular one that it belonged to. And so this whale meat that was being served was from the sei whale, and that's that particular whale is pretty endangered. And so what we're looking at is probably one of these unknowns right here. Right, so if you take a look at the results of this, right? So they were serving it up. And so you can kind of see the minke whale, the fin whales, the Birdie's whale, and here's a sei whale right here. And then so that's most likely where it actually falls onto the actual chart. So when you build a phylogenetic tree, we're going to use homologous structures. And so remember, we always try to do monophyletic groupings. And so sometimes, though, what you'll see some trees that are paraphyletic or polyphyletic. And so sometimes when you look at these trees, right, like a paraphyletic tree, it has a common ancestor and majority of its descendants, but now we're excluding one. So paraphyletic, we're excluding one. Polyphyletic, what you have is a common ancestor, and then you actually grab one from another branch, which it's not part of the same descendant line. And so one of the things why there's so much confusion is because a lot of these researchers, they're actually analyzing and trying to make a closer look. They're looking at various types of stuff, and DNA's genetics is actually playing a huge role in a lot of this. So these trees are actually constantly being rearranged and shuffled around on there. So especially when researchers are learning more and more of the evolutionary relationships to these various taxonomic groups on there. Okay, and so try to get them into so that's part of the reason why a lot of researchers, they're trying to organize them into monophyletic groupings, and also so they're not relying upon traditional morphological taxonomic features that way on there. So if we look at a monophyletic group, right, so here's a monophyletic group right here. So I just kind of use this as an example so you can actually see, right? So here's a monophyletic group, and here's a monophyletic group right there. And then if I did a paraphyletic grouping, right? So I got all the dogs over here, the canids, and then we grab the otter right here, and then this one as well. Mustelids, if you wanted to know, those guys they all have like a really strong scent gland, right? So skunks are actually in there as well. And then polyphyletic grouping. Right, so now we grab one from a different tree, and then on the paraphyletic, right, we excluded one on there. I think I forgot to say that one. And so earlier I just kind of drew some nested clades for you guys. So again, we're trying to look at monophyletic groupings, and then even with all these, there's still further nests that you can actually have in each one of these. So in this madness of red, blue, and green circles right here, what you're looking at are nested clades. So you can draw these nested clades on there. So notice that the canids and the mustelids, those are clades because you got the common ancestor and the descendants. Mustelids and canids are actually nested. That's the red circle. And then if we take the green circle, right? Now we're still talking about a nested clade, a larger clade, and taking a look at what are we taking a look at? We're taking at monophyletic grouping right here. All right. So here's actually a academic genealogy tree. If you watch the earlier intro video that I did, you'll actually my two graduate advisors are on this tree. And my advisor Don Hoyt, he and I forgot who his cohort was that actually came up and drew this particular tree. This is back in the 80s when they drew this tree, or maybe early 90s, I think. And so my other graduate advisor, Stephen J. Wickler, he's on there. He's actually Bill Dawson's graduate student, who was George A. Bartholomew's first graduate student. And there's a lot of names on here. So if you ever gone to a UC, you're most likely been on campus or may have even taken a class with somebody off of this tree. UCI, UC Riverside, UCLA, Schumacher, big University of Washington, Stanford, Colorado State, University, CU Boulder. So a lot of UNLV. So a lot of big name schools on there. Okay, British Columbia, UCB is on there. Okay, so a lot of the big schools, a lot of schools back east as well. And so they're my two graduate advisors. I'm a small twig off of both of those branches. And actually, there's I would have been another branch, another twig. I was looking at going to be James Carnegie's graduate student, and I actually applied and I was accepted into the University of Wollongong down in Australia to be Bill Buttimer's graduate student. But things worked out for the best. I didn't end up going down there. So, but here's Bill Dawson. This is Stephen Wickler. This is my other graduate student, Don Hoyt. So if you're at UCI, all right. Oh my gosh, Al Bennett, Jim Hicks, Ken Nagy these guys are at UCI, UCLA, UCR, UNLV. He does micro stuff now over at UCI in education. So now what we want to take a look at is cladistics. So cladistics now we organizes based upon homologous traits, homologous structures on there. Right? And so what we're looking at is who is most closely related. So we're going to look at what are called shared characteristics. Right? So these are homologous structures. What do you all share in common? And then we're going to look at derived characteristics. Derived characteristics are the evolved characteristics you didn't see before until this particular period of time. These organisms actually have it. So some organisms have what we call shared derived characteristics. Okay? And so shared derived characteristics. So we have apomorphies. Apomorphies are the derived characteristics. These are called apomorphies. Apomorphies are derived characteristics. A plesiomorphy is more of a primitive characteristic. So a characteristic that the outgroup shares with the in-group on there. Okay? So something that C, for example, will be our outgroup. B and A are our in-group. And so a plesiomorphy is going to be a characteristic that C, B, and A have in common. Because C, B, and A are sharing that same primitive characteristic, that's called a symplesiomorphy. So a symplesiomorphy is a shared primitive characteristic. In this case, A, B, and C all share that. Now, apomorphies are the derived characteristics. So B is going to have an apomorphy that C doesn't have. And because B is going to share that same derived characteristic with A, so A and B are going to share that derived characteristic. So there's going to be a shared derived characteristic. That's called a synapomorphy. So a synapomorphy is a shared derived characteristic. What's going to be between A now? A is going to have an apomorphy that B and C do not have. So A has an apomorphy that B and C do not have. So because A is not sharing it with B and C, that's why it's called an apomorphy. Now A and B share a derived characteristic, so that's why it's called a synapomorphy on there. So when you construct a cladogram, you're going to choose the taxa that are the clades. Okay? You're going to determine the characteristics. Oftentimes when you guys get it, you guys are going to get characteristics. And then you're going to determine the polarity of the characteristics, who has it, who doesn't have it. And then we're going to use an outgroup comparison, right? And so the outgroup is always going to be furthest on this part of the chart. Right? And so we're going to group the taxa by synapomorphies, the shared derived characteristics. And we're going to use the rule of parsimony. So the easiest and simplest explanation is going to be the most widely accepted. So if it takes you like 2 different steps to get to that particular point, right? Or you have the option of taking only a single step, you're always going to take the single step. You're always going to take the least number of steps to get to that point. So we're going to construct a cladogram, and we're going to use the outgroup comparison. Each of the taxa goes on the ends of the points. And so we need to have a characteristic chart that's kind of established. And so you guys always have a characteristic chart in order for you guys to actually do this on here. So the taxa go on each of these endpoints. Okay. Our amphibian is going to be our outgroup. It shares characteristics, so it has a symplecomorphy. Right? So there's a symplecomorphy right here with the amphibian, and that's going to be a backbone. The horse, lizard, and the human is going to have a synapomorphy that these 3 share that the amphibian doesn't have. And that's going to have, this is abbreviated as AM, this stands for amniotic membrane. And the amniotic membrane is shared by the horse, lizard, and the human, but not the amphibian. The amphibian still has a water drive. It has to go back to water for reproductive purposes on there. All right. So now we have to resolve this node right here, horse, lizard, and human. So who's going to come next on there? What characteristics do these 3 share that the other doesn't share? Okay, so the way I did this to separate them out, right? The horse and the human, we all have hair. We have mammary glands on there. So the lizard does not have hair. The lizard does not have mammary glands. So this is the apomorphy that horse and humans have that the lizard does not. Okay? This is an apomorph. This is the amniotic membrane is a synaptomorphy that's shared by the horse, lizard, and the human. But this is an apomorphy for just mammals, not for the lizard, not for the amphibian on there. So the question now comes: I happen to put the horse here, and I put the human over here. How do we separate these two? What characteristics can we use to separate the horse and the human? All right, so we have mammary glands, we have hair, right? But we have things, who has something that the other doesn't have, and is it shared amongst others and so forth? So, but if we look at us, right, one of the big things that we do is we walk bipedally, and we also have an opposable thumb. Well, you say that, well, the panda has an opposable thumb, so what? That's actually a bear. That's carnivore. So when we're looking at this is, so we could use either one of these two, right? And the opposable thumb, when did that come about? And that was a more of a later derived characteristic on there. So if we look at bipedal, could a horse be bipedal? That's what you want to think about. A horse could have been bipedal, but now it's a quadruped. Maybe it is more efficient that way. So if you think about this, did quadruped was that first, right? Or was it a biped first? Or which way? Now maybe horses were bipeds like us, and then now they became quadrupeds. But that's two-step. Wouldn't it be easier if everybody was quadrupeds and then humans became bipeds? Right? Ever wonder? So now this takes us back to development, right? And so we're going to use Occam's razor for this. So think about it this way. We start out, right? Once we finally can do what is this called? Oh, what is this developmental sin when you're upright on there? I don't know, reverse seal. I don't know if it's called reverse seal. There's early childhood education, you get that. I forget what it's called. And so, once you get to here, right, you're strong enough now. You can start quadrupeding. Now, before that, you were just like a little steamroller. You just rolled everywhere. Right? But now, what you're doing now, you're on quadruped, right? So now you get up here, you start standing up. Now you're quadruped for a while, and then you slowly start to walk, and you become efficient on there. Does that make sense? So one step going this way, whereas if everybody was biped and then you went quadruped and only humans retained being biped, there's so many other animals that went back to being a quadruped. So most likely it was quadruped, and based upon just looking at us, right? We started out as quadrupeds, and then now we go biped. And then for those of you guys that are going to be parents one day, right? You want your kid to crawl as much as possible. You don't want them walking right away, because when they're quadruped, what they're doing is they're scanning their environment, right? And that helps with reading comprehension. And so normally I would tell you about the story of where my sister, when she was teaching third grade, and she had a kid that could not read very well at all. He was reading at like a kindergarten, first grade level. And this was like, before they finally figured out why he was having a hard time in school. And so they sent him to therapy. He did a lot of crawling, a lot of scanning and things like that. By the end of the school year, he caught up to be reading at a third grade level. So this is why you want your kids to crawl as much as possible. For some of us, you need to get back down and start crawling again. Right? But each one of those, the amniotic membrane, the hair, the opposable thumb, those are synapomorphic. Although this one right here, this is more of an apomorphy. Okay, all right. So these are synapomorphies right here because they're being shared by others on there. All right. So on this slide right here, what you're looking at is vertebrates. And we're looking at all the amniotes. Well, not all the amniotes, right? We have reptile. We have the in group right here are the mammals. We have the duck-billed platypus, which is egg laying. We have the kangaroo, which is a marsupial. They have a really short gestation period. And then we have a placental mammal, right, which has a longer gestation period. And actually, long gestation, you can actually change that to being placental on there as well. So again, here is a little cladogram using the outgroup comparison. So what I want you guys to do is practice drawing this. So on your assignment that you guys have, you have a practice cladogram with some dinosaur characteristics on there. There's 4 dinosaurs. And what you're going to do is you're going to practice putting those dinosaurs on the cladogram in the correct sequence based upon the characteristics. So you have a little chart like this. And then you have another chart with a bunch of different animals on there. I'm going to have you construct a cladogram from that. And I'm going to have you construct a phylogenetic tree on there. And so when you're looking at this, we have a lancelet. This actually already says outgroup. So you know that the lancelet's going to go right here. It's not going to have any of these apomorphies that's going to be shared amongst the lamprey, tuna, and the salamander. So, and oftentimes notice how this is nicely organized for you. Who has the most characteristics in common, right? So you look down at lamprey. Lamprey has one more, so it has a vertebral column. So what you're going to do is you're going to put the lancelet there because you know that's the outgroup. And the lamprey now, it doesn't have a backbone, so that's going to go next. So notice it right here. I put my characteristic right there. The backbone goes right there. And then I come up here to the next one. Here's jaws. If you want to be a predator, you got to have jaws. Lampreys do not have jaws. These are referred to as jawless fishes. They're agnathans. And they're eel-like, but they do have teeth, and they'll grab on. They have a rasping tongue, and that will scrape off scales and tissue and so forth off of there. All right, so I'm going to put jaws right there. and then tuna is the first one that has that, and because it doesn't have anything else, because it doesn't have walking legs, I just put tetrapod instead of four walking legs on there. Then we have the salamander, because the salamander does not have an amniotic membrane, and then the turtle has that because that's a reptile, and then the leopard is the only one with the hair, and so that goes on the very end. So the leopard has the most synapomorphies. The lancelet, right, has no synapomorphies. Right? So each one of these are the synapomorphies right there. Right? So here you can actually see what it actually looks like on the cladogram. So now this time what we're going to do is draw a phylogenetic tree. So using the same group of critters and the same set of characteristics, we're going to draw a phylogenetic tree instead of a cladogram. So if you lose points on the assignments, is because you get them confused, or you draw your tree funky, or you draw your cladogram funky. Make sure that you draw it properly. Right? And then on a test, when I ask you guys to draw one of these things, I've had folks draw it incorrectly. So you should get a 0, but you get a general idea. So you get like half the points for that. So make sure you know phylogenetic tree versus a cladogram. Okay. Cladogram kind of looks like a little check mark thing, whereas the phylogenetic tree looks like a bunch of branches coming off of. So what I found the easiest thing to do is find the organism with the most apomorphy. That's going to be the leopard, and I start backwards instead of building it the other way where we had the lancelet, and then we had the lamprey, the tuna, the salamander, turtle, and the leopard. I like to build it kind of backwards, and so what I like to do is I kind of like to draw it out, right? So as I draw this, right, so here's my next one. Right? So notice you kind of see what I'm doing. I'm kind of going backwards. So notice I have 1, 2, 3, 4, 5, 6, and 1, 2, 3, 4, 5, 6. So now that I have my phylogenetic tree, and I apologize for them not being all at the same line right there. I was trying to clip art or not clip art or whatever. All right, so then I got my leopard, my first, right? There's turtle, salamander, fish, lamprey, lancelet, and which one's our out group, right? And those are the one with all the zeros on there. That's our out group right there. And then what I do is I come back and then I put down my characteristics on there, right? So here's the vertebral column right here, right? So the lancelet doesn't have a vertebral column, but everybody else does. Fish have a hinged jaw, so does everybody else, but the lamprey is jawless. 4 walking legs. I actually wrote 4 walking legs, tetrapod this time. And then we have the amniotic membrane, and what does the leopard have? It actually has a fur on there. So you want to put the characteristics on there, right? These apomorphies on there as well. What happens right here? That's the common ancestor, right? So if I circle all of these right here, what do we have? Monophyletic grouping, right? Here's the monophyletic group. What can we say about the turtle and the leopard? If I draw this circle right here with this monophyletic, these are sister taxa. Okay. And then here's the common ancestor right down over here where the red arrow is. So here's what the tree actually looks like as well. That's the easiest way I found to draw it. We have 2 different hypotheses for this phylogenetic tree on here about sharks and ray fin fish. So we are going to take a look at some sharks, and we're going to look at some ray fin fish on there. So on hypothesis 1, we have the shark as the outgroup, and then we have a ray fin fish over here. And the reason why the shark is out here is because they have cartilaginous skeleton. Ray fin fish are the bony fish. And there are some fish, however, with cartilaginous skeletons on there. Those are chondrostii. They include things like the sturgeon. So bony skeleton, right? So this is one. Now this other one has it where we have bony skeleton, and then we have it where sharks and the ray fin fish are actually sister taxa that branched off a lot earlier. So which hypothesis do you think is more likely to be accurate, and why? That's what I want you guys to think about on there. So earlier we saw a phylogenetic tree with the elephants, and that had unproportional branches on there. But that unproportional branches had the x-axis labeled with a timeline on there. And so that tells us, depending on the length of that particular branch, determine when that particular taxa died or became extinct on there. In this particular one, this phylogenetic tree with its proportional branch lengths, this one, as long as there's some type of description on there, this one happens to be the number of genetic changes for a particular DNA sequence on there. So notice that the mouse and the human and the chicken are all pretty close to the same, but the frog has more changes, the lizard, the zebrafish on there. So that's a model organism right there. Right? Fruit fly also is a model organism. So these are all slightly different. A lot of these genes that we're looking at are going to be those Hox genes that we talked about in the earlier lecture on there. So earlier on, I had mentioned to you guys that, I'm going to medical school. Why do I need to understand this? Well, you guys are looking to help save lives, right? And some of you guys going into medicine, you might discover some particular drug that's going to cure something, but you're not going to get to test it out on people first. So what are you going to test it out on, right? So you're going to have to find a model organism to kind of test it out on. And so, why do we need to understand cladistics, right? Well, it's a good way on how organisms are grouped together. So you might question, why are, this organism and this organism kind of grouped together? Why are they similar? I can see a cat and a dog, they're closely related to each other. But, where did the bear fit into all this? Okay? And so one of the things here is we're looking at for predictions, right? What genes might function in other organisms. And then the big one is how compounds work, right? For drugs and so forth. There was a research project that came about because one of the students, not my students, but one of Huntley's students, was a pharmacy tech. And a patient came in and said, hey, I need some more Xanax on there. And what happened to it? We just refilled it. You can't get well, I had it. I don't know why they had it sitting on their aquarium, but they knocked it over and the pills fell into their aquarium. And so her first question was, what happened to the fish? So this came about an experiment where they looked at how Xanax affected fish. And what they used was Siamese fighting fish, right? The betas, male betas on there. And it was actually pretty telling that. So notice that the same receptors that we are utilizing for the Xanax, also fish have these same receptors. And so here's the thing, right? When we get to the bone structure, I'm going to talk about structures that are in the fish and in the reptile, and then in us, and how they change through that. So there's a lot of these are the big ones right here, where a lot of you guys are going to be utilizing one of these days on there. Okay? And so, but these are why you should have an understanding of cladistics and phylogenetic trees and evolutionary relationships on there. All right. So with that, here's the assignment that you're actually going to work on. Here are the dinosaurs that you guys are going to place on the tree, on this cladogram right here. And then these group of animals right here, here are the characteristics. They're all kind of mixed up on there for you. Pluses, they have it. Minuses, they don't. Zeros, they don't have it. And then pluses, they have it. I want you to construct a cladogram and a phylogenetic tree on there. So if you have any questions, you're confused, you need help, please ask. Shoot me an email, pop into Zoom office hours, right? They will be towards the end of the lab period, and work on the stuff, right? If you have any questions at any time, I'm here to help you, right? So have a wonderful day, and I will talk to you soon. Have a good one. Bye.